Hello, Krishna.
Hi, Bradley.
How are you?
I'm fine. How are you?
Okay, that's good.
Yeah. So, the paper that I was going to write on starch is almost complete. I'll mail it to you tomorrow, by tomorrow.
Okay, that sounds good.
We just have to write. Do we have to, format into a full paper or just an abstract?
This is for what?
For Neuromatch.
Neuromatch. Okay, yeah. Just the abstract,
Okay.
and then
I have written a whole paper with all the references and citations.
Well, actually, yeah. Why don't you send me the whole paper, and then maybe we can submit it somewhere?
Yeah.
Because there's some conferences coming up, and I don't know what there are always conferences coming up, so we can always submit it somewhere like that.
Yeah, we can do it. It's like it's more of you can say cognitive science and neuroscience and deep learning type of thing. My paper is a mixture of all those things. So I'll mail it to you by tomorrow.
Okay. Yeah. Yeah, it sounds good. Hi, Jesse.
Hello.
Hi, Jeffrey.
Hi.
Yeah, so Krishna was just talking about Neuromatch submission, so he's going to submit something. Yeah, so send me the abstract. So for Neuromatch, you need like a 200 to 300 word abstract, and then you'll need a presentation to go along with it. So it's good that you have a paper. You can just draw from that to put the presentation together. And then the paper itself, we can maybe submit to some conference. So let me take a look though. Hi, Susan.
Yeah.
Hi.
Hi.
Can you hear me?
Yeah.
Hi.
Yeah.
Okay, good. I just have trouble with my camera sometimes.
Oh, yeah. So, welcome to the meeting. So, Susan, are you going to present today on?
Yeah, I can. Yeah,
Okay.
I've got it up on my desktop, so I could do that.
All right. So, why doesn't Susan go first, and then we'll talk about the general meeting, and then we'll talk about Hacktoberfest at the end. So.
Okay, so I'll put on present now, or have you?
Yeah.
No,
yeah, the lower right-hand corner is a button present now.
Okay, there's only 3 of you.
Yeah, that's fine. That's fine.
That's okay, just
Yeah.
okay.
Yeah.
Where is this? Present now. My entire screen, sure.
Yes.
There you go. Entire screen. Share. You see it? My screen?
Not
see
yet.
yourself?
Not yet.
Okay. Okay, can you see that?
No.
Not yet.
Let me see if you can try sharing a window as well. That might work.
Oh, okay.
So if you share the window and where it is, if it's in one of your. Hi, Ujjwal.
Share entire screen.
Hello, Faisal.
Hi.
Yeah, it doesn't want to do that. Chrome wants to share contents of your screen.
Yeah, say yes.
So how do you share the window?
If you click on lower right-hand side, present now, it should come up your entire screen or a window.
Okay, present. It just says present now.
Yeah, go ahead and click that.
Okay. And I should do a window?
Yes.
And see if that works. Okay. There. Oh, that's better.
Okay. There we go. I see it now. All right.
You can see it?
Yeah. Yeah, I see it.
Okay. There, is that even better?
Yeah, I can
Hopefully?
see it now. Yeah, it's good.
All right. Okay, so this is my thesis topic. Optical coherence elastography of livestock tissue. And I'm working with biomedical engineering at the University of Manitoba. And anyways, so I'm doing this. It's been used for. Oh, does somebody want something?
Oh,
No. Anyway, so this is good for cancer margin detection, enhancement of histology images, and actually getting histology image of living tissue. And you can check the changes in healthy tissue, and you can watch how cellular processes work, and you can study embryo development with this. So for elastography, we're measuring the viscosity and elasticity of the material. And viscosity is the resistance to flow, and elasticity is a measurement of how a substance reforms after it's stretched. And then you have some measurements are stress, which is force over area and measured in pascals, and strain, which is a ratio of deformation length over original length, and then Young's modulus, which is stress divided by strain, and is also in pascals. So the tissue is non-linear in many ways, and it's anisotrophic, thixotrophic, and I said non-linear, and has hysteresis. So anisotrophic means it's not the same in every direction, and thixotrophic means that it is initially hard to move. It's like mud or clay, and it's normally given as ketchup, where you're trying to get ketchup out of a bottle and you're shaking it, and all of a sudden too much. So all of a sudden it moves. Optical coherence elastography, it's based on optical coherence tomography, and that uses infrared light with an interferometer or camera to obtain images that are 2 to 3 millimeters in depth. So it has it's not a very deep image, but it certainly goes into tissue, in particular, say my 2 millimeter diameter egg, and skin. Anyways, optical coherence elastography is measuring the deformation of tissue and how it bounces back, of course. And we're determining the viscosity and elasticity of the material. And basically, you're taking an image that before, during, and after deformation. And you're also doing simulations of the material so that you can compare a simulation of the actual measurements to get a better idea of what's going on with the material. There are two common setups used with optical coherence tomography. There's a spectral domain optical coherence tomography, and the first figure here, and the swept source or Fourier domain optical coherence tomography, or OCT, in the second diagram here. And I'm going to be using the second setup to start with, at least. And this is of some well, made up samples, known samples. So this is your OCT, or your image of of these silicone samples. And this is soft material and stiff material. And then this is the strain. And then this is the elasticity. Yeah, with soft and stiff material. And lovely images of, or not images, here's some diagram of the Young's modulus, which is stress over strain, of various body parts. And I hope to be doing more things more like a brain and lung and ground substance. Anyway, there's quite a range with brain at one end of this and bone at the other. And what happens to living tissue when you change its environment? This is breast tissue, and it goes from a nice annular conformation to something that's moving, depending on the stiffness of the matrix or the ground that it's on. And this is epithelial cells, or endothelial, pardon me. These are endothelial cells, and if you have flowing blood and pressure, you get this type of cell. And if you don't, you get this type of cell, and they've known to round up and die if they don't have pressure. This is a stem, and if you have a stiff substrate, you get bone. If you have a viscous substrate, you get cartilage. And if you have a soft substrate, you get a fat or adipose tissue. So methods of measuring the stress in tissue, you have to perturb the tissue in some way. So you can compress it mechanically, or you can do that with ultrasound, or you can use stress, which is hypertonic is too much salt and hypotonic is too much water. So you can stress it that way. And you also embed magnetic beads in the tissue and pull on them, or you can add tissue with a tube, or you can use air puff indentation. You may see that at your optometrist office if they've done that test with you. You might be able to do water puff. There's photothermal, where you heat it up. And they have something called nanobombs. They've actually used this. They're explosions by lasering nanoparticles. Now, the bottom ones in black here are kind of invasive, I would say. So probably just using the top three. Types of mechanical manipulation. So there's your compression, optical coherence elastography, and you can compress it with a needle in one spot. This is the one in pink here is the magnetic bead one, and then ARF is, I should really put that in there, acoustic radiation force is what that is. So that's an ultrasound. And then you can, dynamic indentation is where you sinusoidally indent it. And then there's air puff and your photothermal. They don't have the water absorption one here. It's the block diagram of how to do this. So there's compression, swept source loading, acoustic loading, etc. And then to detect these methods, indentations. You have speckle tracking, sensitive detection, Doppler spectrum detection, and image compression or comparison. Sorry. The image comparison is just letting the tissue do what it's going to do and then comparing how it moves. A window at 1300 nanometers is one of the better ways of measuring things with infrared light because it has a better penetration through water at 1300 nanometers. Also at, what is that, 1100 or so, there's also one, and then at 1600 nanometers, but 1300 is the best, and that's what Dr. Reed has. So that's initially what I'm going to be doing. There's a diagram of the setup in Dr. Sharif's lab. It uses an interferometer, and it's actually 1310 nanometers with the middle frequency. And this is the actual setup, and it's well, basically set up and ready to try things. I'm going to be altering it somewhat. And here's a holder that I made up. I may or may not use it depending on how well it works. Anyway, this is a piezo electric actuator with a little piston, and you put the tissue in here and you compress it. And it's flexible because you can just compress it, or you could do a sinusoidal compression or other things. An experiment. This is the actual experiment here with actually gathering live data, and then this is your model. So you're checking what your actual data is compared to a mechanical model. And the mechanical modeling is difficult. Anyway, this is what you get when you do this. This is the actual optical coherence tomography image, and then this is, it says the local displacement. So this is how it's displaced when compressed. No, this is how it's displaced when it's sorry. And then this is the actual strain. So you measure the strain. And then there's an image that's reconstructed, and it's using our inverse map and a model to construct it, and this is the elasticity. Quite well, because along this column here you have the actual measurements. This is the optical coherence tomography, and this is the displacement, and then this is your optical coherence elastography image, or the elasticity. And on this side, this is the simulation. And down below we have the blue line is the actual experiment, and the red line is the simulation. You can see they're quite close. And so you've actually checked your measurements by doing the simulation. And optical coherence tomography does not injure tissue. At least it appears not to be. This salamander was put in the optical coherence tomography laser when she was an egg, She grew up normally and laid normal eggs. And then this is an optical coherence tomography image of an egg at 1300 nanometers with a swept source. And swept source means it's plus or minus 55 nanometers, so it goes below 1310 nanometers by 55 nanometers and then boots up to 55 nanometers above 1310 nanometers. Anyway, this is an interesting image. It was taken in Dr. Sharif's lab by his adjunct and the references. And you have some questions.
Well, thank you, Susan. That was a very good talk. I have a couple of questions. Does anyone else have questions before I ask mine? Let me revisit a few slides. I like to do it that way. Can you go up?
Yeah.
Let's see. I'm interested in the sort of the tissue stiffness graph that you showed. Is probably up in like slides 5 or 6, maybe.
Yeah, this one?
Yeah.
Mm-hmm.
So that's a nice graph. So this is reported range of Young's modulus in tissue. And so this is Young's modulus, which is measured in pressure, and then structural hierarchy, which is sort of like the smallest things you can find to the largest. So you have cells, basically, or cell components, structures, and then organs. And so you said that brain is the softest tissue that they tested there.
Yes.
Bone is the hardest.
And the bone is the hardest. Yeah. But airway mucosa, that's, I ran into actually a paper on horse students. So they're actually measuring these things with the optical coherence elastography. So, not sure the motivation behind the horse view, but anyways, yeah, horse.
Yeah. So it's interesting that like these constituent parts here, you think about like a brain or a bone, what is making up this tissue. And so you can see that like you have, with bone, you have more things that have a higher Young's modulus, like elastic, keratin, collagen, and brains have things that are less. Right. And so that's how you get this sort of measurement of the entire tissue versus the components.
Yes, so you can see bone and calcified plaque. Well, basically calcified, so yeah.
Yeah. I'm trying to think of the other one I wanted to discuss. How about slide 11?
Uh-oh, sorry.
Okay.
Can you? I'll just see if it that way. Sorry. Anyways, so slide 12. There. Oh, this one?
No, the one up, the one before it.
What about? Oh,
yeah,
okay.
That one. Okay. So yeah, this is interesting in light of some of the things we talk about in the group. So this is, I think we've talked about this before. This is an undifferentiated stem cell at the left. And so what you're doing is you're basically introducing mechanical stresses to the cell. And those stresses are going to result in some differentiation cue for the cell. So like a lot of times what they'll do is they'll take a bunch of cells and they'll plate them on a media, they'll have like when they do cell culture, they have a plate, and that's a medium, of some, or a surface of some type. And then they'll put media in the plate with cells, and the cells will attach themselves to the plate. But the plate is patterned usually in some way. And so you can get like a pattern on the plate that induces different forces. You can also do things like subject them to viscous forces in a fluid so that you can do things. You could put them in a bioreactor, but you can also just introduce forces at a regular interval with like flows and things like that. And so the idea is that the cell gets exposed to these cues, which are environmental cues, and it triggers some gene expression program to move. So in here, well, in this case, this is in a, I guess this is in the body. Well, this is like a section from where they are in the body, right? So cells are in, yeah.
Endothelial cells in a vessel.
Okay, in a vessel.
And yeah. And so if you actually have blood flow and pressure, you get the bottom configuration, and the top configuration is endothelial cells without. And they're actually different. They're expressing different genes.
Yeah.
They tested this. So
yeah. And so, and they also have, I guess they have different configurations with respect to one another. So they touch each other, and they have all these cues from their environment, and it results in different gene expression programs. So you have some genes that are upregulated, some that are downregulated. People write papers where they actually use antibodies to stain out different genes that are being expressed. So, I mean, there's a lot of stuff going on. And then, of course, a bunch of genes being co-regulated in some ways, usually a way you get different cell types. So if you go to the next slide.
back
to the 11. Yeah.
It's interesting because my daughter did some work in a lab, and the people in the lab studying endothelial cells said they had to clamp it off the blood vessel off so that or their endothelial cells would die on them. Ptosis.
Yeah.
So
it is that does happen a lot actually when you get this. So what happens in this undifferentiated stem cell is you get this what they call multipotency, and so you get like a number of different fates that you can achieve with this stem cell. So like this, in this case, you have bone, cartilage, and fat. So you can have one of those fates, and it just depends on the cues. But of course, in the process of differentiating, sometimes the cells die. Sometimes they become cancerous, so they just kind of get dysregulated. And so that's basically what happens when the cells are go from the stem cell to a differentiated cell. They can take on different fates, but it's not entirely stable. There's some slop. It can go off the rails, or it can go to these fates, or it may stay undifferentiated in some, in a lot of like cell culture conditions. Sometimes, the cells will differentiate at different rates. Like sometimes, a little bit of stimulus will result in some cells becoming differentiated, but other cells will remain stem cells, and you have to.
So, okay, I'm about tissue engineering here.
Yeah.
You have undifferentiated stem cells, and you're trying to create tissue. You're giving them the right cues, especially people think of giving them the right chemical environment. But if they differentiate slightly wrongly, they can become a cancer cell. And so it's very important to put them in the right mechanical environment as well.
Yeah. It's always tricky if you're doing like cell culture to get the mix right, and a lot of times it takes a lot of trial and error to see good results. Like your medium won't be very good, or you need to use chemical cues to induce things, and so it's It's a
combination of chemical and physical, and you need to get them right. If you're replacing this chunk over here because somebody lost a chunk of, say, muscle, you've got to be aware of both the chemical and physical environment that it's supposed to go into. and how it, the precursor stages, but even where does muscle start from? It's undifferentiated stem cell, and then it has to go through this process. And a lot of that is mechanical as well as chemical.
Yeah.
So, yeah, it's complicated.
Yeah. Well, thank you for that. I guess we don't have any
I have a
questions. Oh, you do? Okay, go ahead. Oh, you have a question, Krishna?
Am I being audible?
Yes.
Yeah. So, like type 1 muscle fiber and type 2 muscle fiber might have different, Young's modulus, and people who exercise, athletes have, harder muscle cells. So, will it bring some ambiguity when we are, categorizing muscles on basis of their, you can say, mechanical characteristics? So, muscles of someone who lift weight would be have high, you can say high, would be more stiff. So will it be, get some create some ambiguity while you can say when we are categorizing them?
You'd be able to see the different muscle fibers, or if you can't, it's not a good thing. You should be able to see the individual muscle fibers and see the, they say, the Young's modulus. You'll have to look at the strain with this technique, and then have a known substance surrounding it so that you can see the stress. So then you can get the Young's modulus. So yeah, you see both of those types of fiber and see the difference between them when you're doing this technique.
Yeah.
Thank you.
Yeah.
Hope that answered that. Okay.
Any other questions? Well, that was very good. Thank you for that, Susan.
Okay, I'll stop sharing it.
Okay. Actually, if you could send a link to the slides, if you have them, I can put them on Slack for our people who aren't here. They can see it.
Okay. Well, I could send you the PDF version.
Yeah, that'd be good.
It's not as long. So
Okay.
did I stop sharing the screen?
Yeah, you did.
Great.
Okay.
Okay.
All
All right. I'll even turn off my video.
Okay.
Yes.
All right. Thank you. And this is we had a couple of comments, but they were all share slides. Thank you. So thank you. So the next order of business is the annual update. So tomorrow we have an annual meeting for OpenWorm, and the meeting is at 12 o'clock Eastern time in the U.S. So that's, I can't remember what that translates into UTC. I think 4 o'clock p.m. UTC. So if I actually invited Ujjwal and Mayuk to give little short presentations on their things, on their summer projects. So if they can make it, then they can present. I'm going to give some time for them to present on their materials. And I think it'd be good to expose and introduce the DevoLearn to the rest of the OpenWorm group. I don't think I think we did some sort of like presentation at the beginning of the summer with what we want to do, but we've not shown the finished product. So this is very timely to get people introduced, a wider audience introduced to this. And so our senior contributors are going to be the people who are at this meeting. So we have people ranging from people who do like neural modeling to people who are doing like, informatics, we have programmers, we have all sorts of people who are doing different things in OpenWorm. And so it's going to be, I think, interesting for them to see what we've been doing here. We often don't get to share our things because we're in our own Slack channels and we're doing our own things and in our own meetings, and it's hard to get. We don't really have as many cross project get-togethers as we used to. So it'll be, I think, informative for them. And then what I wanted to do today, let me share my screen actually. What I wanted to do today is to go walk through this presentation that I'm going to present on behalf of the group to the senior contributors. So this is the latest DevoWorm logo. I reworked this recently. This is the 2020 update for the OpenWorm Foundation. Here's the GitHub repository, and here's the website. So our logo's gone through a couple of iterations over the years. So this was the first logo from 2014 to about 2017. And there was this logo from 2017 to 2020. It has like 3 topical areas, which, we've kind of drifted away from some of these, but this is basically what we've done work in the past. And so then this is the new logo. It's a little bit cleaner, and so the DevoWorm group, this is the slide I use to kind of describe how we do things, as opposed to some other research groups that do that collect their own data primarily, or they do like computational work. So we're affiliated with OpenWorm, where they want to build the world's first digital C. elegans in an open source manner. We have a lot of things that we do using simulation analysis and visualization. So we're interested in development of C. elegans and other creatures. So we're interested in, we're going to talk about axolotls. We're going to talk about Basilea. And so we'll talk about those as well. We do work on different organisms. If you go to the website, you'll see in the publications that there are different organisms that we do, as well as computational work. And then we have, here's DevoZoo. I have to change I think I have to change this link, but this is basically the third part of this. So we have openly available primary, secondary, and tertiary data sets. And this involves like segmented microscopy data or raw microscopy data, literature mining, and public repositories. I think we had a question. Oh, yeah, thanks, Jesse. Nice logo update. Yeah, so let's see. So that's, I'll double check these slides if we're a percent, but I think those are the right links. So this is developing the virtual worm. This is a talk that we presented at Virtual Worm 2020, which was because of the pandemic. It was organized by a postdoc out of Baylor Medical Center, and he did a series of talks on C. elegans. And so it was mostly biological research, but we had a spot and we presented on a bunch of things that involved both sort of data science and theoretical work. And so one of the things we talked about in that talk was this idea of biological models avoiding spherical cows. So this is an old, there's an old joke about physicists. Physicists can model anything as long as it's a sphere in a vacuum. And so it's a joke that means that you're oversimplifying your system by assuming certain things about the system. So in this case, you're thinking of a cow, but you're thinking of it as a sphere. And that's, of course, not how cows exist in the real world, but it's an easy thing to model. And so people, that would be the joke. But we also, in development, we deal with spheres. We deal with embryos that are spheres, and they turn from spheres into these highly asymmetric organisms. And this is originally something that Dick Gordon talks about in some of his books, but I think it's worth highlighting here that if you oversimplify a biological system in terms of modeling and in terms of data analysis, that it can not be a very good analysis or model. And so but this is an example of some of the work, and this is some of the work we presented at Neuromatch. These are embryo networks, and so this is a paper that was published in 2018. I don't think the larger open-worm group has seen this work very much, but this is what we've done here is we've taken an embryo and we've generated these networks, and you can look at different cells and the relationship between the cells in terms of space by plotting out a network. And so this was presented at the Neuromatch conference, which was also something organized in response to the pandemic, where we were able to show that you have these spatial patterns within an embryo that exist throughout development. So you start at a one cell or a 2-cell embryo, and the cells start to divide and differentiate, and then form these patterns that in the form of tissues. But you see a lot of patterns amongst the cells in terms of their position, in terms of their distance from one another, even before you start to get tissues. So you start to get asymmetries early, you start to get other things going on early before you really start to get tissues forming. And so this is what this work was. And you can actually use these kind of embryo networks to capture a lot of features in biological development. So in the C. elegans, we have C. elegans that's newly hatched. And so we're drawing a network around that phenotype. We can also capture things like in the mouse blastocyst, we can capture differentiation of different tissues by labeling the different cells as to what tissue they belong to, and then plotting them out on one of these circular graphs and showing the connections or the connections based on distance between cells. And we can see this sort of modularity emerging. And so this is a potentially powerful tool for analyzing development. We've really kind of laid out this method in this paper, and we're planning to do another paper as well. And it'll be interesting to see where that kind of work goes. And this summer, of course, I think Mayuk did some work on network models, and we did some in 2018 during GSOC. So it'd be an interesting area for further investigation. Of course, we've also done a lot of work on deep learning and quantitative morphology. So this is C. elegans embryogenesis, where we take an embryo, we find the centroids of cells, and then we also find the edges of cells, so we can create segments of the cell body. We can create centroids of the cell body. We can plot them in a coordinate system and analyze the embryos and create networks like we just showed. We also have looked at Basilea cell colony morphology. So this was work done by Ujjwal, Singh, and Asmit Singh, where they segmented the bodies of Basilea cells in a colony. This was also work done by Thomas Harbeck, where he segmented the cells and then measured different landmarks on the cells and calculated out a lot of that. And so there are a lot of things that we've been doing in this area. It's very exciting work. And to this end, we've done research, but we've also done education. So in this case, we've hosted a course called DevoWorm ML, and this was hosted in 2019, at the end of 2019, so about a year ago. There's a GitHub repository for these materials. And this course was basically like a semester-long course. It brought together machine learning and developmental biology. So, we did a series of topics in machine learning, but with a developmental flavor to it. We did blog posts, papers, presentations. We looked at innovation in machine learning, and then we innovated in the educational component by featuring this on Google Meet and by distributing the materials. And this actually has the potential to engage with the rest of the open-worm community and around the data science theme that we've been talking about for a couple of years. And so, but also we've gone in the direction of research, and this is sort of the culmination of a lot of our efforts, and it's called DevoLearn. This is a project that has involved a number of people. this last summer, we did a lot of stuff in terms of Google Summer of Code, in terms of setting up this platform. So we have our DevoLearn platform, which is hosted in a GitHub organization. Within that organization, we have a program called DevoLearn 0.2.0. This is something that's hosted on both GitHub and PyP. It's an analysis program for people to take some raw data, some raw image data or movie data, and segment the cells in different ways using deep learning techniques. Then we have a couple of other components. We have species-specific models, which are characterized by DevoWorm AI, which is a platform. And we also have Heroku apps, which are apps that host different species-specific models that also use deep learning and other techniques to analyze images of cells. And so we have one for C. elegans. That was the product of the 2019 GSOC program. We also have the Basilea model, and we're looking for additional models to add to this library. Finally, we also have DevoZoo, which I mentioned before, which is a source of secondary and tertiary data. So once we get these, images analyzed, that creates a secondary data set. And then you can create a tertiary data set by annotating those data that are extracted from the images. And then those are all hosted at DevoZoo. And so those are hopefully be helpful to education and researchers alike. And so DevoLearn has been a group effort. DevoWorm AI has been one component of that. And so you can see here's a screenshot of the sort of the front page of DevoWorm AI. And this, you click through and you can do a lot of things here. You can do analysis. There's some educational component as well. This is a screenshot of the DevoLearn repository. So we have these, this is the PyP front page. So this is like, there's an example notebook in here that kind of lays out what you can do in DevoLearn 0.2.0, how to run the program, and so forth. Finally, we've done a number of papers that have been published in the last year. I'll put two here for people to see. The first one is Raising the Connectome, which was a paper that was single-authored. It was based on the earlier work on developmental connectomes, which actually is related to the network research that was shown earlier. But it basically considers a C. elegans connectome and how it develops from a single cell that's differentiated way back at like 200 minutes of embryogenesis, tracks the growth of that connectome, and then considers how the connectome sort of wires itself up and then starts to produce behavior. So it's a really interesting paper. It considers a lot of things that really haven't been, it reviews the literature, but it also kind of introduces a model for thinking about the developmental connectome. Then the second paper is this Towards a Digital Diatom. So this is the work on Basilea, which is a type of diatom. And this is a paper where we use deep learning and some biomechanical techniques to look at Basilea and think about it as more of another digital organism modeling effort. And so this has been accepted to the book Diatom Gliding Motility, but this is the bioRxiv version. Finally, we have some ongoing projects. One is this Basilea psychophysics. So we've done the Basilea work with the Basilea morphology, but now we're moving into psychophysics, which is this notion of being able to quantify behavioral outputs. So in this case, we have a human perceptual system trying to distinguish between dot arrays of different orders of magnitude. And so you can see that you have this array of dots, and if you compare the array of 10 dots versus the array of 20 dots, it's easier to distinguish between the two than an array of 110 dots versus an array of 120 dots. To say that there's a difference, it's a little bit harder to do. And so we hypothesize that Basilea has similar, not necessarily in sensing dots, but in sensing maybe concentrations of things, or their movement dynamics that we can look at. And so feedback from movement and other things like that. And so this is one thing that we're working on. We're doing research on. We're trying to figure out how to best represent the organism. Do these behaviors represent an autonomous intelligence, or do they simply mimic intelligent behavior? So we're actually asking a question that we've asked in OpenWorm a lot, where we look at like a simulated worm and we ask, is that real or is that just simulated? Here we're looking at real organisms and we're seeing if we can figure out whether it's really like intelligent behavior, whether it's just kind of a mechanical type of behavior. We're also doing a lot of stuff we're moving into axolotl embryos as well in terms of modeling microscopy images. And so in this case, we're trying to create a map of this embryo these data have been generated using a special type of microscope where the embryo is flipped on its axis, so it rotates. And the rotational movement actually can give us a full view of the entire surface of the embryo. And so we're trying to figure out a way to create a visualization of this. And this will be included into the DevoLearn repository as well once we're done. So thank you to our contributors for 2019-2020. I've listed you in this list. I don't know if I missed anyone. I was trying to think of who has been attending our meetings regularly, but I think I have everyone. I also have our Google Summer of Code students for 2019 and for 2020. So thank you for your contributions. I would like to say, in terms of the DevoLearn slide, that we also have a lot of data science tutorials that we're trying to build. And we've already gotten a couple of contributions there, but also we're trying to build out some of the other parts. We have other parts that we're thinking of adding, like theory building and that. And so that'll all be part of DevoLearn as well. So that's basically what I have for the annual report. If you can think of anything else, let me know by tomorrow morning. I know it's hard to think of anything that I missed. I probably have a better beat on it than anyone, but so again, I'm going to be presenting that tomorrow, and hopefully people at DevoWorm will think it's great and inquire more about what we're doing here. And again, Ujjwal and Mayok are invited. We have 30 minutes, so maybe we can reserve maybe 10 or about 10 minutes for both of them to say something about their individual GSOC projects. But I think we have a lot of stuff already there. Okay, well, that's fine, Ujjwal. You don't have to attend. I just wanted to give people the opportunity. Yeah, I'm sorry.
I think that's a really, I appreciate the presentation, even as someone is sort of a flapper and I come here all the time, but just seeing all the stuff together is really helpful. And I think it might be nice to link that to, I don't know if people learn directly, but having access to that for people who are coming to the lab and be able to say, hey, here's what we did. Yeah. I think that will help a lot of stuff and bring a lot of things together. So it's good to see that. Yeah.
And, if we, we'll probably have some follow-up from other people. It's usually the way to, we don't ever get to present the stuff to other parts of the organization. So it should be, I think people will at least have links and they'll be able to see things and go beyond what I'm just, presenting there. And it's, it'll be a good time. We have, every year when we get together, we always talk about like the data science initiative. Last year we talked about doing that and that didn't really go anywhere, but maybe this year we can get more momentum behind us on it. We'll see. New link. Okay, thank you. That's the new link to DevoZoo. So that's, the index. I have to Thank you, Ujjwal, for reminding me. I'll change that in the slide deck. So the last thing I want to talk about quickly before we go is Hacktoberfest. So I think Mayuk may have mentioned this, and also Krishna, about Hacktoberfest. So Hacktoberfest is something that is hosted on GitHub. Usually different organizations participate, and you basically, during the month of October, you can contribute to a repository. And I think if you make a number of contributions, I don't know if GitHub gives you like credit for something, or sometimes the organizations give out swag or something like that. It's a way to get people involved. Yeah.
Like in Hacktoberfest, there is a like kind of a rule, like if you have successfully pushed 4 or more pull requests to some organization or something, so they provide you some stickers and T-shirts. So this is like how it works.
Okay. Yeah, I knew they had some rule about, yeah, so it's 4. Yeah. So it's, if you commit a certain number, make a certain number of commits during the month, you get like some, GitHub can give you like, where, the organization, we haven't really thought about like doing it in DevoWorm or doing it in OpenWorm, which we might give people for that. But it's definitely worth sort of promoting. And so I think we've talked about promoting it on DevoLearn. And so what I can do for that is I can put out a blog post at the beginning of the month, and then maybe we can get some people to promote this, throughout social media. Maybe people will come and, okay, thank you, Ujjwal. I promoted DevoLearn in my college groups. So this is one thing, like I think we've already promoted DevoLearn a little bit in terms of social media and the blogosphere. But, to have like, say, well, Hacktoberfest, you can get special acknowledgement of your commits or something like that. It might, drive some contributors to the platform. We don't know yet. So this is the Hacktoberfest logo. The way I have it here on this README is, welcome to Hacktoberfest. Details coming soon. In the meanwhile, check out our DevoLearn repositories and our DevoLearn AI resources. Want to participate in Hacktoberfest at DevoWorm? Look at our issue board for group meetings. So I have the group meetings issue board, which we'll need to like update a little bit. Or look at the contribution guidelines for DevoWorm. So we have the DevoLearn contribution guidelines, and you can contribute a data science demo. And even, I think Susan actually mentioned that she had some material she might contribute to that. We really want any kind of data science oriented or data analysis oriented stuff you have. So if you have something, you can submit it. I will create good first issues and Hacktoberfest issues in the Devo. Oh, that's good. Thank you, Ujjwal. Yeah, we can create Hacktoberfest issues. I don't have a Hacktoberfest tag, but we can make one. If you go into the issues and you go into the labels, you can create a label, and it's Hacktoberfest. And we can label those with good first issue and Hacktoberfest labels. I think the good first issue is already there, but like the Hacktoberfest one is, you just type Hacktoberfest with a description, and that's the tag. And you can use that for different issues. So so let's go to the issue board just to show you what I mean. So this is a label presentation, but we can put other labels. If I look at this one, say for example, when I look at the labels over is hard to see, but they drop down like this, and then you have your good first issue. You have all these other labels, and then you can also make new labels. And so that, I think, using the labels, I think will guide people to the appropriate tasks. So I just want everyone to know that, because October starts at the end of the week. So getting into the swing of that would be good if we want to do something for that. And then it goes throughout October. And next week we'll maybe talk a little bit more about how to organize for that. It's people are busy, and but it's just I think it's a nice way to see what we can do during the month to get people involved. Well, thank you for coming to the meeting, and thank you, Susan, for presenting on your materials. And Krishna, I look forward to seeing your paper if you can send it to me. And if you're interested in the Neuromatch submissions for abstracts, that is due Friday. So if you have an abstract you want to present at Neuromatch, you can submit the abstract. It's about 200 to 300 words. I know Jesse and Krishna are already doing something for that, so it's just a matter of okay. And then any update on the boring billion? Yes, thank you for reminding me. So the boring billion paper is on. I need to send Ujjwal the notes that I made on our meeting from last week. We did a meeting. We talked about like what is involved, and I need to make some issues out of that, but I'll send them to Ujjwal. We did have a meeting last week, and we did kind of lay out some first steps, but I need to figure out what we can do. Yeah. Okay. Thanks for meeting. Have a good week. Oh, wave papers. What are the wave papers? Okay. Oh yeah. So I think I already sent him something, but I'll double check. Okay. Talk to you later. Bye.
Yeah.
Okay, bye.